All right, thank you so much. So as Lindsay mentioned, I will be the moderator for this session. So I will be introducing the speakers, and then at the end of the session, I will be asking the speakers your questions that you have typed into the chat. And feel free to type them whenever they occur to you during the talk. You don't have to wait until the end. And then I will get to them in the order that they're put in there. All right, so our first speaker today is Dr. Polina Casolo. Polina completed her PhD in neuroscience at the University of Oxford, working under Professor Stephanie Craig, where she showed that cortical control of striatal dopamine release was possible via cortical cholinergic connections. Currently, she is a senior postdoctoral research fellow with Professor Helen Bateup at UC Berkeley, where she studies how genes implicated in syndromic autism spectrum disorder and Parkinson's disease impact the dopamine system on molecular, structural, and functional levels. The overall goal of Polina's research is to advance our understanding of the core organizational principles of dopamine neuron function at the molecular level and ultimately establish how molecular changes in dopamine neurons govern behavior. So with that, I'll hand it over to Polina.
Thank you, Paul. Let me just share my screen. Can everybody see it now? Okay, fantastic. So thank you, Paul, for a very kind introduction, and to ViDA organizers for my invitation to speak today. I would like to share an exciting story about mTOR signaling in dopamine neurons that I have been working on over the past few years. So the dopamine system that we all love and study is actually a very complex beast. The cell bodies residing in the substantia nigra pars compacta and ventral tegmental area project to their targets via medial forebrain bundle and form topographically and functionally distinct neural circuits. So at the simplest level possible, nigrostriatal pathway is critical to motor function. Mesocortical projections modulate cognition, while mesolimbic terminals sought to orchestrate motivational processes. Now, as a field, we came to appreciate that distinct subpopulations of dopamine neurons can be distinguished based on their soma position in the midbrain, their forebrain projection topography, gene expression, and many other parameters, which in turn translate to distinct functionality. However, we still know very little about molecular processes which orchestrate dopamine neuron function, and this is something that really piques my interest. So, with dopaminergic neuromodulation controlling so many aspects of behavior, from movement to cognition, the dopamine system actually is a fantastic candidate for studying molecular control of cells and neural circuits that are important to behavior. And so in this talk, I would like to convince you that understanding of mTOR signaling could be one way of linking molecular processes to behavioral aspects in these very important cells. But furthermore, I hope that you will see how such studies also reveal some of the fundamental molecular principles which control functional output of dopaminergic system in a cell-autonomous manner. So, mechanistic target of rapamycin, mTOR pathway, is ubiquitously expressed, highly conserved signaling cascade which integrates intra- and extracellular signals to regulate processes that are integral to cell survival. mTOR signaling comes in two distinct flavors, mTORC1 and mTORC2. These two signaling complexes are formed by distinct complement of obligatory binding partners, Raptor for mTORC1 and Rictor for mTORC2. Functionally, mTORC1 in particular is responsible for lipid and protein synthesis and actively inhibits autophagy, while mTORC2 conventionally thought to control cell proliferation and survival. mTORC1 and mTORC2 in turn also have distinct upstream activators and downstream effectors. TSC1 TSC2 heterodimer serves as a negative regulator of mTORC1 activity, keeping it in check via Rep GTPase. In turn, status of mTORC1 signaling can be examined by looking at phosphorylation of its downstream target S6, while for mTORC2, activity can be read out based on phosphorylation of its target AKT. So in this talk, I will give you a brief introduction to mTOR signaling in dopamine neurons. Then I'll talk about how loss of TSC1 and ensuing constitutive mTORC1 activation impact dopaminergic system. I will touch upon whether mTORC1 and mTORC2 control over dopamine function is equivalent, and then I will summarize and conclude. So what evidence do we have to begin with that mTOR has anything to do with dopamine neurons? Well, first, both in vivo and in vitro studies in mechanical or neurotoxin lesion models of Parkinson's disease show that acute changes in mTORC1 signaling actually increase dopamine neuron survival. Second, both mTORC1 and mTORC2 signaling in dopamine neurons are engaged by common drugs of abuse and actually have been implicated in addiction. Thirdly, we can detect phosphorus 6 indicating mTORC1 activity in midbrain dopamine neuron somas at baseline. The final piece of the puzzle comes from neurodevelopmental disorder Tuberous Sclerosis Complex, which is caused by loss of function mutations in either TSC1 or TSC2 genes, which causes constitutively active mTORC1 signaling because negative control supplied by TSC1-TSC2 heterodimer is lost. So tuberous sclerosis patients have very high rates of autism and a wide variety of neuropsychiatric conditions. So much so that a specific classification system called Tuberous Sclerosis Associated Neuropsychiatric Disorders, or TAND, was developed. So these aberrations spanning behavioral, psychiatric, and neuropsychological functions have something very interesting in common. They all thought to involve dysregulation in dopaminergic function. And therefore, aberrant mTORC1 activation could be important in causing disturbances in dopamine neurotransmission, which in turn cause neurobehavioral difficulties in TSC. To formally test the possibility that dopaminergic dysregulation caused by constitutive mTORC1 activation contributes to TAND, we induce dopamine neuron-specific deletion of TSC1 gene using .irs-CRE mouse line. Loss of both functional copies of TSC1 causes constitutive mTORC1 activation as negative control from TSC1-TSC2 heterodimer that normally keeps mTORC1 signaling in check is lost. So in this experiment, we specifically opted for conditional TSC1 gene deletion to examine cell autonomous changes in dopaminergic circuits and also assess whether mTORC1 constitutive activation exclusively in dopamine neurons is actually relevant to TAND phenotypes. So one possible readout for high mTORC1 activity is phosphorylation of its downstream target S6. Phosphorus 6 antibody staining reveals significantly higher mTORC1 activity in the midbrain of TSC1 knockout animals compared to wild-type controls. And quantifications on a cell-by-cell basis plotted as cumulative probability reveal that both SNC and VTA dopamine neurons have significantly increased phosphorus 6 levels following TSC1 loss, and also consistent with mTORC1 hyperactivity phenotype, also show a dramatic increase in their soma size. So how would this impact intrinsic properties of these cells from electrophysiological perspective? And we also wondered whether that would extend to their morphology. So interestingly, hypertrophic features caused by constitutive mTORC1 signaling also extended to the dendritic arbor. Pictured on the left are representative examples of reconstructed wild-type and TSC1 knockout dopamine neurons located in SNC and VTA. These dopamine neurons filled with biocytin during whole cell patch clamp recordings and reconstructed with MRIs show a dramatic increase both in the number of dendritic branches that is determined by Scholl analysis and in the total dendrite length. So we wondered whether as a result of this hypertrophy, our cells could show changes in intrinsic excitability. And so actually consistent with somato-dendritic hypertrophy that we observed, we also see pronounced hypoexcitability both in SNC and VTA dopamine neurons. So pictured on the left side are representative examples of action potential firing evoked with a 300 picoamp current step. And by injecting current of increasing amplitude in the whole cell patch clamp setup, we observe a significant increase in the rear base current, so the current at which dopamine neuron fires its first evoked action potential in TSC1 knockout cells specifically. Also, by constructing excitability curves, we see that at any given current step, up to 400 picoamps, TSC1 knockout dopamine neurons fire fewer action potentials compared to wild type. So overall, this means that TSC1 knockout dopamine neurons would be less likely to fire action potentials in response to weak depolarizing input, yet would still be able to burst fire with sufficiently large depolarization. And hence the contrast between tonic and phasic firing is likely still preserved. But as most people, or probably everybody in this meeting knows, that action potential firing does not directly translate into dopamine release events in the target regions. So we also examined electrically evoked striatal dopamine release using fast scan cyclic voltammetry. So in this experiment, we uncovered a significant reduction in peak evoked dopamine release throughout all 7 sampling sites across dorsal and ventral striatum. Now, I would like to draw your attention to the fact that the magnitude of change was substantially higher in dorsal striatum, sites 1 through 6, compared to recordings performed in ventral striatal regions in the nucleus accumbens core. And so overall, averaged across all sampling sites in dorsal striatum, we saw approximately 60% reduction in evoked dopamine release and roughly 15% reduction in the nucleus accumbens core. Well, then we wondered whether actually with such severe impairments in TSC1 knockout dopamine neurons, would they degenerate over time? So remarkably, this phenotype was actually very resilient in the face of aging. Recordings performed in the 22 months old animals showed an overall decrease in evoked dopamine release that's actually expected with age, yet 60% reduction in dopamine release from dorsal striatum in TSC1 knockout mice was actually still present and very robust. Of course, having found a substantial reduction in evoked dopamine release, we wanted to make sure that neurotransmitter was available for release in the first place. And so we harvested the entire striata to examine protein levels of tyrosine hydroxylase, TH, which is the rate-limiting enzyme in dopamine synthesis, and VMAT2, responsible for packaging synthesized dopamine into vesicles. So consistent with our observations from immunohistochemistry experiments, both on midbrain and striatal sections, we saw a significant increase in striatal TH expression with Western blot, which suggested that dopamine synthesis, in fact, was increased with constitutive mTOR1 activation. To confirm that possibility, we further examined the total tissue dopamine content in dorsal and ventral striatal regions with high-performance liquid chromatography in collaboration with Sarah Threlfall at the University of Oxford. Again, we saw a significant increase in total tissue dopamine content both in dorsal and ventral striatum. So together, these findings presented us with an unexpected conundrum. Dopamine was indeed there, but dopaminergic axons actually failed to release neurotransmitter upon stimulation, and that was not helped by increasing extracellular calcium or the amplitude of electrical stimulating current. So in the FCV experiments, we observed reduced dopamine release probability from TSC1 knockout slices, despite conclusive evidence of increased dopamine synthesis and content from Western blot and HPLC experiments. And therefore, we hypothesized that actually ultrastructural changes could underlie release deficits and turned to examine dopaminergic axon terminals with electron microscopy in collaboration with Peter McGill and Natalie Duig at University of Oxford. So here you see representative electron micrographs of dopamine axon terminals labeled with immunogold in dorsolateral striatum and in the nucleus accumbens core. We examined these specific regions as examples because of distinct release impairment profiles I pointed out to you a couple slides ago. Dorsal regions presented with profound functional deficits, while nucleus accumbens core terminals had milder phenotype. So first, as you can already probably appreciate, EM data showed that hypertrophic changes also extended to dopamine axon terminals following TSC1 loss. Yet, increase in profile area compared to wild-type control was actually a lot more pronounced in dorsolateral striatum compared to the nucleus accumbens core. Secondly, EM data also revealed a significant reduction in the vesicle number in TSC1 knockout terminals across both regions. Yet, given the substantial difference in the size, this actually translated to profound reduction in vesicle density in dorsolateral axon terminals and comparatively smaller reduction in vesicle density in the nucleus accumbens core. We also looked closely at vesicle parameters that would directly impact their ability to fuse with cell membrane and release their content. Specifically, we examined vesicle distance to perimeter and how far any given vesicle was within that terminal, how far away it was from other vesicles. And this we termed intervesical distance. Our data showed that following TSC1 loss, both vesicle distance to perimeter and intervesical distance was significantly increased compared to wild type. Furthermore, once again, these changes in vesicle positioning were actually a lot more pronounced in dorsolateral striatum compared to nucleus accumbens. Another way to look at intervesical distance is to actually assess vesicle clustering properties. And in this analysis, we found that in dorsolateral striatum specifically, vesicles in TSC1 knockout terminals were significantly less clustered, so the red bars, compared to wild type, the gray bars. No such change was seen in the ventral side. So overall, our observations are consistent with the fast scan-cyclic voltammetry data, where release impairments are actually much more pronounced in dorsal than ventral striatum, and suggest that ultrastructural reorganization at the axon terminal level with constitutive mTOR1 activity causes dopamine release deficits. Next, we wanted to zero in on possible molecular effectors that mTOR1 is recruiting to control structural and functional properties of dopamine neurons. One way in which mTOR1 could do this is actually by controlling protein synthesis that it does via its downstream targets S6 and 4E-BP1. To make it possible to examine how TSC1 loss impacts translation, we crossed our wild-type and TSC1 knockout.RS Cree mice to Cree-dependent GFP-L10 mouse line, where a major ribosomal subunit is labeled with GFP. Using brains harvested from these animals, we performed translating ribosome affinity purification, TRAP experiments, which allow us to isolate actively translating mRNA. So in this setup, midbrains are harvested and processed to isolate ribosomes with mRNA fragments that they're currently translating. Dopamine neuron-specific expression of GFP-L10 ensures that magnetic bead capture of GFP-expressing ribosomes collects mRNA coming exclusively from dopaminergic neurons. The rest of mRNA fragments are actually isolated and captured as background signal. Both background and dopamine-specific mRNA gets purified, undergo library preparation, and sequencing. So sequencing data was analyzed with the help of John Blair, former graduate student in the lab, and our TRAP experiments performed in wild-type and TSC1 knockout dopamine animals uncovered the total of 508 differentially translated genes shown as a heat map. The overwhelming majority of transcripts actually had upregulated expression, which in fact is consistent with mTOR1 activity, leading to massively increased protein synthesis rates. But we also saw downregulated expression in a few genes. The volcano plot on the right shows some of the top hit targets which meet cutoff criteria for fold change. So preliminary analysis such as top heat go terms and ingenuity pathway analysis show significant changes in mitochondrial gene expression, autophagy, and several other cellular functions that we are going to pursue for target validation. And so specifically from this list of differentially translated genes, we created a manually curated list of genes with known function that are expressed in CNS and midbrain, and also have probo antibody commercially available. These genes include those important for autophagy processes, translational control, mitochondrial health, synaptic function, dopamine neuron identity, and axonal transport. So the first step will be to verify that translational changes can be detected at the protein level with Western blot and immunohistochemistry. And for the validated targets, we will further pursue in vivo functional studies using shRNA and CRISPR approaches. Now, I would like to go back to the idea of linking molecular processes to behavioral phenotypes. Dopamine neurotransmission plays an important role in many different behaviors, and I will not bore you with the phenotypes that we actually did not find. But I would like to tell you about very interesting and remarkably specific behavioral deficit we consistently observed in TSC1 knockout mice. With the help from Linda Wilbrecht's lab at Berkeley, we set up an order-based reward learning task that is known to be sensitive to dopamine, and for simplicity, I will call it the 4-choice test. In this task, a food-deprived mouse is learning an association between a particular order and a small piece of palatable food it wants to eat.
Paulina, I think
Paulina, you are muted. Just from the beginning of this task where you talked about collaborating with Linda Wilbrecht.
Sorry. Okay. Am I back? Okay. So the task. Linda Wilbrecht helped us to set up the task, her lab. I will call it for simplicity 4-choice task. In this task, a food-deprived mouse is learning an association between particular order and a small piece of palatable food it wants to eat. So this schematic shows you that the task is performed across 2 days. On day 1, acquisition day, mouse learns the initial food order pairing. And on the second day, the contingency that mouse learned on day 1 is actually reversed. And this we call the reversal phase. So the picture on the right shows a mouse who correctly performed the digging response and is now consuming the palatable food. I would like you to take note that order location actually changes randomly on each trial, and therefore task cannot be completed based on spatial cues. So what do we see in this task? Trials to criterion measure is used to evaluate when mouse successfully learned the task and is set to 8 out of 10 trials performed correctly. So what you can see is that during acquisition learning on day 1, wild-type and TSC1 knockout animals actually perform very similarly. However, once order food contingency changes in reversal phase on day 2, TSC1 knockout animals on average are actually showing significant impairment in their task performance. Specifically, they take significantly more trials to learn the new food order contingency, and on average, they make significantly more errors. If you look closely at the cumulative reward plots for individual animals, you can see that by trial 20, all of wild-type animals figured that odd food pairing has changed, while TSC1 knockout group could go for up to 40 trials before earning their first reward. Examining types of the errors more closely, we also see that errors TSC1 knockout mice make significantly more are in fact preservative in nature. This means that animals keep going back to the previously rewarded order, even though it's no longer providing them with the palatable food piece that they want. Now, perhaps the most dramatic illustration of this preservative effect can be seen in the order choices mice make during the reversal phase. So in the first few trials, wild-type mice realized that food reward is gone from the initially rewarded order, and they stop choosing it and quickly start to explore other options. In stark contrast, DA-Tsc1 knockout animals continue choosing the no longer rewarded order, showing cognitive inflexibility, a phenotype that is very common in human tuberous sclerosis cases, and in fact in autism spectrum disorder. Now, to demonstrate that mTOR1 hyperactivation is indeed instrumental to dopamine release deficits and cognitive inflexibility phenotypes that we observe, we examined whether constraining mTOR1 signaling in the context of DA-Tsc1 loss can ameliorate those changes. So we did this using a genetic approach, once again to ensure manipulation specificity to dopaminergic neurons. mTOR1 requires obligatory binding partner Raptor to form the complex, and hence reducing Raptor availability would constrain mTOR1 activity even when DA-Tsc1 negative control is lost. So in this case, losing one copy of Raptor gene indeed led to a reduction in mTOR1 activity, as illustrated by reduced phosphorus 6 levels in Raptor het animals compared to Raptor wild-type animals. Remember that both of them are still on DA-Tsc1 knockout background. So what did it translate to functionally? Did it help? To examine functional impact of heterozygous Raptor deletion in the context of DA-Tsc1 loss, we once again performed fast-scan cyclic voltammetry experiments. And we saw that loss of one copy of Raptor gene on DA-Tsc1 knockout background improved evoked striatal dopamine release across all of the sampling sites. And averaged across all of the sampling sites in dorsal striatum, DA-Tsc1 knockout Raptor het evoked dopamine release was roughly 50% higher compared to DA-Tsc1 knockout Raptor wild-type animals. So we see prevention of dopamine release deficits. Does it also prevent behavioral phenotypes? So we further then examined the performance of these animals in the 4-choice task and asked, did it improve? Again, you can see that during acquisition phase, the two experimental groups are actually equivalent in their performance. However, in the reversal phase, DA-Tsc1 knockout Raptor het animals in blue now outperform DA-Tsc1 knockout Raptor wild-type animals in red, so our original knockout animals, who on average take significantly longer to learn the new food order contingency and on average make significantly more errors. On the cumulative reward plots, again, you can see that DA-Tsc1 knockout Raptor wild-type animals in red cluster out to the right, meaning that they're slower to abandon their initial learning and begin exploring other options that would earn them food rewards. In terms of error types, DA-Tsc1 knockout Raptor het animals now actually make significantly fewer preservative errors on average compared to DA-Tsc1 knockout Raptor wild-type mice. And from order choices in reversal phase, hopefully you can see that loss of one functional copy of Raptor gene on DA-Tsc1 knockout background now enables these animals to realize very early on that the previously learned strategy no longer yields the desired rewards and explore other options. So you can see a precipitous drop in their choices of initially rewarded order. Now, I would like to briefly summarize the data that I just showed you. Loss of DA-Tsc1 leads to loss of negative control of mTOR1 signaling, which renders it constitutively active. As a result, we observe profound somato-dendritic hypertrophy in dopamine neurons, both in SNC and VTA, which in turn leads them to become hypoexcitable. Hypertrophic phenotype is also observed at the axon terminal level in striatum. These ultrastructural changes in turn cause a dramatic decrease in evoked dopamine release, possibly via reduction in dopamine release probability, actually despite increased total tissue dopamine content. And behaviorally, this translates to intact initial contingency learning, but subsequently causes cognitive inflexibility phenotype in the reversal learning phase. Both reduction in evoked dopamine release and cognitive flexibility can be prevented by constraining mTORC1 signaling by removing one functional copy of Raptor gene. Now, in the remaining time, I would like you to tell a follow-up story to the Raptor availability manipulations. A couple slides ago, I showed you that losing one functional copy of Raptor gene allowed us to constrain mTORC1 hyperactivation following TSC1 loss, which in turn improved evoked striatal dopamine release and performance on reversal learning task. It should come as no surprise that losing both copies of Raptor gene renders mTORC1 signaling completely suppressed. Basically, with Raptor gone, the complex can no longer assemble and maintain itself, and consequently, mTORC1 signaling is completely abolished, as evidenced by lack of phosphorus sex signal in midbrain dopamine neurons. Again, we're using that IRS-CRE line to manipulate gene expression specifically in dopaminergic cells. Now, we were pretty surprised when we examined evoked striatal dopamine release to find that complete suppression of mTORC1 activity in homozygous Raptor knockout animals on TSC1 knockout background was actually as detrimental as constitutive mTORC1 activation in TSC1 knockout mice. You can see that these transients are not different from each other. And so these data actually suggested to us that midbrain dopamine neurons are exquisitely sensitive to mTORC1 signaling status, and too much or too little mTORC1 are bad news for dopaminergic neurotransmission either way. And so we wanted to follow up on why this observation was true. In this project, we asked what happens to dopamine neurons when mTORC1 activity is completely suppressed. Now, if you recall, mTOR comes in two distinct flavors, mTORC1 and mTORC2, which play actually distinct functional roles. And so we wanted to find out whether mTORC1 and mTORC2 arms of the mTOR signaling pathway play, in fact, equivalent role in controlling dopaminergic system. Now, we already saw the data where mTORC1 signaling is constitutively active following loss of TSC1. Unfortunately, an equivalent manipulation that would render mTORC2 arm constitutively active is not possible, but both mTORC1 and mTORC2 signaling can be abolished following loss of their obligatory binding partners. And so in this series of experiments, we now took advantage of Raptor and Rictor conditional lines and cross those with that IRS-CRE driver line to achieve dopamine neuron-specific suppression of mTORC1 in the case of Raptor and mTORC2 in the case of Rictor. So what did we find? Homozygous loss of Raptor gene caused complete suppression of mTORC1 signaling as evidenced by lack of phosphorus-6 signal in Raptor knockout midbrain. And the soma size in these dopaminergic neurons, as you can hopefully appreciate, was also reduced. And when we reconstructed biositin-filled dopamine neurons from both SNC and VTA of the Raptor knockout mice, we further observed a profound reduction in their dendritic complexity that was apparent just by eye. Now, Scholl analysis revealed a significant reduction in dendritic branch number with constitutive mTORC1 suppression, both in SNC and VTA, and Raptor knockout mice also had a significant reduction in the total dendritic arbor length. What happened to morphology in the case of Rictor? So with homozygous loss of Rictor gene and ensuing mTORC2 suppression, we saw small reduction in phosphorus-6 level actually in the Rictor knockout mice, which suggested possible crosstalk between the two complexes. Yet overall, dendritic profiles of these Rictor knockout dopamine neurons appear to be quite similar to wild type. At least you couldn't cluster out them by eye. However, Scholl analysis of the reconstructed neurons actually revealed an increase in the number of proximal dendritic branches that was specific to SNC dopamine neurons, and that nonetheless did not cause an overall significant change in the total dendrite arbor length. With such profound morphological changes as caused by loss of Raptor and constitutive mTOR1 suppression, we wondered what would happen to those cells electrophysiologically. So we examined intrinsic excitability of these dopamine neurons. Interestingly, and exactly the opposite to mTOR1 constitutive activity, Raptor deletion rendered both SNC and VTA dopamine neurons hyperexcitable, which was in fact consistent with the hypomorphic morphological features. Dopamine neurons with complete mTOR1 suppression had significantly lower Rio-base current, meaning that they fired their first evoked action potential more readily compared to wild-type cells. However, constructing excitability curves further revealed something very interesting. Up to a point, Raptor knockout cells were indeed more excitable and fired more action potentials at a given current amplitude. Yet very distinctly at 200 picoamp step, both SNC and VTA Raptor knockout cells tended to go into depolarization block and spike no more, suggesting that these dopamine neurons would actually have a compromised ability to burst fire in response to a large depolarizing input. Was the same true for mTOR2 suppression with Rictor? Well, interestingly, no. In turn, our Rictor deletion did not appear to have a significant impact on SNC dopamine neuron intrinsic excitability, even though we did see a significant increase in the number of proximal dendritic branches. Yet, constitutive mTOR2 suppression did render VTA dopamine neurons more excitable, as evidenced by significant decrease in the Rio-base current in VTA Rictor knockout cells. Increased intrinsic excitability in the VTA Rictor knockout dopamine neurons could also be glimpsed from excitability curves and actually is consistent with previous reports in addiction literature. So what then happens to dopamine release? So to complete the picture of changes in dopamine neurotransmission caused by mTOR manipulations, we then assessed evoked dopamine release with fast scan cyclic voltammetry. This time, site sampling experiments actually revealed a significant and ubiquitous reduction in evoked dopamine release in dorsal and ventral striatum of Raptor knockout animals where mTOR1 activity is suppressed. So in this case, averaged across all sampling sites in dorsal striatum and in the nucleus accumbens core, we observed approximately 50% reduction in evoked dopamine release. And in Rictor, well, that was not the case. In contrast, Rictor knockout animals actually showed site-specific modulation in evoked dopamine release changes. In this case, the overall reduction in dorsal striatal dopamine release was actually driven by sampling sites in the ventral portion of dorsal striatum that receive proportionately larger input from VTA. The dorsalmost sites, sites 1 through 3, were actually unimpacted and are predominantly innervated by SNC projections. The reduction in evoked dopamine release in the nucleus accumbens core, which again receives predominantly VTA inputs, was also small but significant. So overall, it was about 10% reduction in peak evoked dopamine release levels in the impacted sites. Where did it come from? So to answer that question, we examined the possibility that neurotransmitter availability could impact dopamine release and explain the phenotypes we see. Now, with Western blot analysis of striatal tissue for the Raptor knockout animals, we saw a significant reduction in TH expression and DAT, dopamine active transporter. And thus, with reduced TH availability, which in fact compromises dopamine synthesis, there appears to be a homeostatic downregulation in DAT expression, which possibly enables better functionality of what little dopamine is left. We further observed a significant increase in the ratio of phosphorylated form of DAT to total DAT, suggesting that what little transporter is still around was actually recruited to do its work of taking up extracellular dopamine back into presynaptic terminals. Significant reduction in total tissue dopamine content, both in dorsal and ventral striatum, was also confirmed with high-performance liquid chromatography on Raptor knockout mice that we performed in collaboration with Bradley Roberts at University of Oxford. We further observed a significant decrease in DOPAC levels, which is a primary metabolite of dopamine, and suggests that lack of dopamine indeed stems from compromised dopamine synthesis, and therefore severely and ubiquitously compromised dopamine release with constitutive mTOR1 suppression, in fact, stems from lack of neurotransmitter to be released in the first place. In the case of Rictor manipulation and mTOR2, we did not see significant changes in either TH or DAT expression, and neither did we see changes in total tissue dopamine or DOPAC content. And therefore, compromised dopamine release in striatal sites receiving predominantly VTA innervation actually does not stem from reduced dopamine production. So in this final slide, I would like to summarize our findings regarding mTOR impacts on dopaminergic system. Overall, I hope that I was able to convince you that mTOR appears to be a critical regulator of midbrain dopamine neuron structure and function. We can manipulate mTOR1 activity levels bidirectionally and hence reveal that mTOR1 exercises bidirectional control over dopamine system, whereas for mTOR2, we can only suppress signaling of the complex. So what happens? With mTOR1 manipulations, we see bidirectional change in somato-dendritic morphology that is robust and observed both in the SNC and VTA. In case of mTOR2 suppression, we see very specific increase in proximal dendrites in SNC cells. Electrophysiologically, mTOR1 suppression leads to hyperactivity or intrinsic hyperactivation of SNC and VTA cells, which fire action potentials more readily. In turn, with mTOR1 constitutive activation, we see that those cells become hypoexcitable. In case of mTOR2, we actually see very specific change in intrinsic excitability in the VTA dopaminergic cells, but not SNC. And finally, with mTOR2 suppression, we seem to observe deficits in dopamine release, small but significant, that predominantly observed in ventral parts of dorsal striatum and in ventral striatum in the absence of changes in TH or dopamine synthesis. In turn, both mTOR1 and mTOR1 hyperactivation and mTOR1 suppression cause very similar phenotype in terms of evoked dopamine release, but they stem from fundamentally different reasons. In case of mTOR1 suppression, TH expression is reduced and dopamine synthesis is reduced as well, which basically means that those cells have no neurotransmitter to release. However, in case of mTOR1 constitutive activation, TH expression and dopamine synthesis are increased, yet structural reorganization at the axon terminal level, which renders dopamine vesicles less ready to be released, renders, what's the word that I'm looking for? Renders dopamine release compromised. So the final slide, I hope I showed you that mTOR signaling is active in dopamine neurons and seems to be a critical regulator of structure and function of these cells. Given the big impact on dopamine cellular function, it probably has relevance for diseases in which mTOR is deregulated. Both mTOR1 and mTOR2 inhibition have effects on dopaminergic system, with mTOR1 overall having greater impact on structure and function. And finally, and this is a piece of data that I probably enjoy the most, both overactivation and inhibition of mTOR1 can be detrimental to dopamine neuron function, but actually via different mechanisms. And with that, I would like to thank all of you for your attention and listening to this talk. Give shout out to fantastic collaborators and generous funding sources. And specifically, I would like to say thank you to Kamran Ahmed, who's done a lot of work on this paper and is just a brilliant scientist. And thank you.
All right. Thank you so much, Paulina, for that very interesting and very good talk. We have a couple questions, and we have a little bit of time for questions here. We started your talk a little late, so I think we can go a little bit into the next talk for questions. So we have one here from Priscilla Ambrosi. She says, thank you, Paulina. Can you speculate on the differing mechanisms underlying acquisition and reversal based on your data? Why does unlearning require more dopamine release in dorsal striatum than learning?
That's actually a very good question. Thank you, Priscilla. Gosh, that would be a pure speculation because I have absolutely no data to back this up with. But I don't think it's so much about the dopamine availability per se, but it's perhaps about the differences between phasic and tonic signals that dopaminergic system would provide to downstream circuits that are important for learning. One possibility is that the contrast is not quite as significant. And therefore, animals may not be getting quite the same level of RPE, if you will. And so they may not, I mean, they probably realize that it's not there because you can clearly see them getting really frustrated. They like start to jump a lot and they expect the reward that they're looking for to be there and it's not. But then it's like a light switch going off. At some point, as soon as they start to explore other options, they actually very quickly learn the task. So there is something about this ability to abandon what you have been doing before and try something else that's really compromised. I think it yet remains to be determined how the decrease in dopamine release that we observe causes this specific phenotype.
And then here's another question from Lindsay that's along the same lines. Given this perseverative activity that's shown in the constitutively active mTOR mice, do you suppose that this might be a mechanism relevant in addiction and drug-seeking behavior?
Yes, I think so. And this is something that I am hoping to pursue in the future. So basically trying to understand how recruitment of mTOR signaling by drugs of abuse then causes the behaviors that result in addiction. There isn't much literature on the subject yet, unfortunately. So we know that it changes the status. So, for example, work from Eric Nestler's lab shows that morphine administration changes mTORC2 specifically in the VTA cells, and then that's what drives the addiction. And if you stop it, that breaks the addiction circuit. But the exact mechanisms by which this happens yet remains to be explored.
Has anyone shown that in addiction, the size of dopamine neurons is increased or decreased?
So decreased, at least specifically for morphine, it's decreased. For other drugs of abuse, I don't think that there are, at least nothing that comes readily to my mind that looked at those parameters.
And finally, I had a question about the vesicles in the axon. You showed that with this proliferation of mTORC1 activity, you get a corresponding increase in overall membrane that leads to these larger cells, but vesicles are also made of membranes. So why are there fewer vesicles? You would hypothesize, if anything, more vesicles, right?
Yeah, that's potentially true. So we don't necessarily know, and this is something that we're going to pursue from the TRAP data. We don't think that mTORC1 hyperactivation leads to increased protein synthesis across the board. We see very specific changes in certain aspects of protein synthesis, but not others. And so why overall decrease in vesicle number? I'm not entirely sure. One possibility, of course, is that we are looking at a snapshot at a given axon terminal. And so given that that axon terminal is now massive compared to the wild type, those vesicles could just be the same number overall, but distributed much further away from each other. And so we see the apparent decrease in the vesicle number that in fact might stem from the fact that they're just not as abundant, basically. So they don't have to be enclosed in the same small space. And so we don't count them as frequently. How that would be possible, I think we still yet to find that out.
Maybe there's time for one more quick question. Do you see any alterations in spine morphology with these various knockouts? This is from Robert Oliver.
Spine morphology in medium spiny neurons, or?
I think in dopamine neuron spines. Then the spines on dopamine neuron dendrites. They do
in
spines.
dendrites. I see. I was thinking axons, so I was trying to figure that out.
No, in the dendrites.
Yeah, in the dendrites, so we didn't do reconstructions on the dendritic spines on the dopamine cells, so I don't know. But that's a very interesting question, possibly. What we can see, though, is that it's basically impossible to be blinded to which condition you're reconstructing, because the cells with increased dendritic arbor are really, really spidery.
Yeah, that was a very dramatic phenotype.
Very much so.
Yeah. All right. Thank you so much, Polina.
Thank you.